So thanks for joining me. My name is Cindy Haynes. I am an associate professor in horticulture and love talking about propagation and seed, and we're talking a little bit about seed today. A little bit of an overview of my presentation. There are 4 main topics that I'll discuss today. I've considered this as a more in-depth discussion of germinating seed. So we've got to talk a little bit about viability and dormancy first off, before we dive into some of those environmental conditions and how do we store seed as well. I'm going to throw a lot of vocabulary at you, and hopefully you have the handout as well, which shows you a few slides and kind of goes over a little bit of that vocabulary that we're going into today. But first, I have to start off with, because I'm a teacher, I have to talk about the different parts of a seed. And when we're talking about a little pre-packaged plant, it's important to know that this is a tiny little plant. There's a tiny little embryo in there with a tiny stem and leaves and root. And so there's certain conditions that we need to make happen precisely to make them, our seed, germinate. These tiny little packages of plants are also perfectly designed so that they can store well and transport well and keep for longer periods if kept in the right conditions. We're trying to get them to break open. We're trying to get them to germinate, and germination generally means in seed germination that this radicle emerges. So all it takes is for the root to emerge, and then it's considered germinated in most instances. When we start with seed, we want to start with viable seed. And viable seed means that it's ready to germinate, that all parts of that seed are still viable. They're still good to go. The embryo hasn't dried out. There wasn't a crack, and the cotyledons have already started to break down. So everything's ready to go. Sometimes we have examples of old seed that have been kept for a really long time and still remain viable. And if they're close to the ideal conditions for that particular species, they can remain viable for a really long time. I think they just opened the seed vault this week and added more seed to them. The seed vault near the Arctic Circle is a perfect place to store seed for a long time. We also have a seed research and storage facility here in Ames. It's not quite like the seed vault, but they maintain a lot of different types of seed, particularly brassica seed and amaranth seed. One of the ways they maintain viability in their seed collection is by constantly growing certain aspects or certain lines of seed and then harvesting new seed from that, so that it's never seed that's kept for more than 10 or 12 or 50 years. The seed are always kind of rotated, and we make sure that we're growing them and harvesting and maintaining viable seed. How long does seed last? That depends on the species. Generally with vegetable seed, this is the seed we try and keep year after year. We have anywhere from a year to 5 years where it actually remains viable. So some things to think about is seeds like onions are only going to remain viable for about a year. Other things like sweet corn, we might get 2 years out of them. And then things like pepper or tomato, we might get anywhere from 3 to 5 years of viability. This also assumes that they're maintained and kept in the proper storage conditions, but it's one of those things that we want to think about. Viability also changes over time. So seed that is sold to you in one of these little packets that you can get at the store is only able to be sold in the year that it's marked. So seed for 2022 is only sold in 2022, and after that, the viability has the potential to go down. And so most seed suppliers are not allowed, it's not legal to sell it afterwards. So if you're keeping seed for longer than that, its viability is probably going down as well. So it can go from anywhere from 90% viability to something less. And how do you know? Is you do a germination test. So this is pretty simple. If you're keeping seed from year to year, just take 10 of the seed out of the package or however you're storing them, place them into a damp paper towel, wrap that paper towel up and put it in a plastic bag, someplace that it's going to remain moist. The bag doesn't have to be sealed, but you want it to stay moist. You're going to put it in a warm location. My grandmother did this on top of the refrigerator because refrigerators always stayed warm. Any warm location will do. It doesn't have to be under lights. It doesn't have to be in a window. We just want to maintain it around 70 degrees. In a week to 2 weeks, you check it. You check, you open it up, you gently unwrap the paper towel, wet it if it needs more moisture, and see what's actually germinated. Remember, all it needs is the root to emerge or the radical to emerge to know that it has completed or is it starting germination. Out of those 10 seed, if you get 7 that germinate, you have a 70% germination rate, and you have fairly good viability on your seed. This really helps you to determine how many seed you need to sow, because after 5 years in some seed, you might have to sow twice as much just to get what you need, because the germination may go down or viability may go down to 50% or less. Seed are sold a number of different ways by local suppliers and national suppliers, and these are some of the terms that you should know as well. Seed are sometimes treated, sometimes they can be brightly colored, bright fuchsia or teal or green or purple, very unnatural colors, and they're often treated with a fungicide, and they're given these unnatural colors so that you know that they are treated. When a supplier goes to the effort of treating that seed with a fungicide, it's a great clue that this particular seed may have problems rotting if it stays in soil that remains too wet or germination media that stays too wet. So think about that when purchasing seed. You can also purchase seed that is not treated. It's organic, that is not treated. But if it also is available in a treated form, that's a good clue that you're going to have to really maintain those proper germination conditions to make sure it doesn't damp off or rot fairly quickly as it's germinating. You also sometimes get pelleted seed, and you're seeing this picture of someone holding seed that is, I think it's lettuce seed or carrot seed that is not pelleted and pelleted. And pelleted seed means it's just coated with a biodegradable coating. This coating breaks down fairly quickly in warm, moist conditions, which most seeds like, but it just makes it a whole lot easier to sow the seed. Pelleted seed are more expensive, much more expensive, but really helpful when you're trying to sow very small or tiny seed. Petunias, sometimes begonias, like I said, lettuce, carrots, those really super tiny seed. It's really hard to grab just one seed and place it where it needs to be, but pelleted seed is just the right size. Still small, but easier to sow, and makes it easier for you in kind of lining them out for germination as well. So it looks a lot better, and they often germinate fairly quickly too. Some seed have a lot of appendages or a lot of excess material on them when they're harvested. So like marigolds often have this appendage or wing that helps it fly, and they might be detailed. So that little tail might be removed. So you might see that term in purchasing certain seed. And then some seed, to make it even easier to sow, are sold as a seed tape. This is an example of seed tape. It kind of feels like tissue paper, and there's little seed in each of these little rows, and they're at the proper spacing already. So this is a salad greens mix of different seed that are already kind of embedded into this biodegradable paper in a sense. So you make a furrow, you line the seed tape out, and everything's and cover it up and then water it, and it's all planted at the proper spacing, ready to go. I love seed tape with kids. And you don't have to spend a lot to get seed tape, and you can even make your own with seed and toilet paper in a whole roll of toilet paper if you want. But it's something that makes it a little more expensive, and certain seed are available this way, which is helpful with certain audiences, especially little fingers and little people. Seed clusters are when the seed that form, there's no way to break them off into individual seed. They kind of form as a cluster. Beets are very commonly sold as a seed cluster, which is why you have to sow beet seed and then come and thin, even if you do an excellent job of spacing them initially, because there's often a kind of a seed that are kind of fused together in a cluster. And then finally, I'll mention prime seed a little bit later. This is, we think of it as pre-germinated seed, or they've been treated in some way to kind of kickstart them, to improve the efficiency of germination. Also a way that some seed are sold. It's an excellent way to start seed in poor conditions because they've already kind of got a good start, and often germinate very quickly. But it's not the best if you want to store seed for any length of time because they've already started part of that process. So I'm going to stop and ask a question of you. Here's another term that you may have heard of when talking about seed, and it's called true to type. So have you ever heard of the term true to type, and what does it mean? And if you could take just a second or two and type that into the chat. What is true to type? Going to give you a few more seconds. I'm looking at the chat a little bit here, Alicia, to see. Okay, last call for those typing into the chat. Excellent. I think a lot of you are getting it. Some of you don't know, and some of you have heard of this one before and you're getting it. And true to type generally means, if I can advance. There we go. Means that the seedling will actually germinate, grow up, look like and produce fruit and flowers that are very similar to the parent. This is sometimes open pollinated seed or heirloom seed, so we can grow them from succession. So you can harvest the seed from this particular fruit, like an apple, grow that apple, and then from seed, and it will produce seed that are true to type, or to much like that parent. This doesn't work with apple. It doesn't work with a lot of our fruit crops, and it certainly doesn't work with hybrid seed because we might get something totally different or something even just a little bit different than what we started off with. This is an important term to know because where you're getting your seed might be important to know if it's a hybrid seed that you shouldn't save the seed from, or if it's an heirloom seed where you could save seed from. Okay. My son, when he was little, always wanted to grow apple seed from the apple as well, and they will germinate. You can produce a tree from this. It's just probably not going to produce the apple fruit that he loves so much and that always wanted to eat. So we've talked a little bit about viability. Now we need to talk about something that will improve germination, and that's something called dormancy. Dormancy is something that's preventing germination in a seed. And different species have different requirements to overcome dormancy, but there are two big categories that we might see. And you need to know if you're going to be an advanced seed starter. So the first is stratification and scarification. So stratification is an instance where that embryo is not fully developed. It has to undergo a winter or a cold period or some sort of environmental period to fully develop to say, okay, I'm ready to germinate. This happens with seed that are growing or native to temperate areas, so that when they fall from the fruit or into the ground, they know that there's a winter coming that they must survive first before germinating. Scarification isn't necessarily an embryo development or physiological issue or physiological dormancy. It's more of a physical dormancy. So it's got a hard seed coat that needs to be broken down, chipped, cracked, damaged in some way so that water can kind of get in. And there's a couple of different ways this can happen. It can happen with repeated freezing and thawing over the winter, or it can be something that happens where the seed is actually ingested or eaten by an animal, maybe in the fruit, and then either gets regurgitated or pooped out the other end. So whatever it kind of goes through its digestive tract, that kind of breaks up that seed coat. Stratification, the term actually comes from when we would stratify seeds several hundred years ago in layers of sand and kind of cool, moist sand, and give it that cool, moist temperature as a kind of a storage technique or a mimic some sort of winter. If you are harvesting seed from outside, from trees and shrubs, they almost always need some sort of stratification period. And it may be a month, it may be 2 months, it may be 3 months. And in this graph, you can see that if you harvest a bunch of seed from acorns or from maples and you germinate or try and germinate them quickly, you will notice that a few will start to germinate, but generally nothing happens for at least a couple of months or close to a couple of months. If you store them or stratify them in kind of a cool, moist environment for 2 months and then try to germinate them, you often get better than 75–80% germination, and generally within a couple of weeks. So it greatly improves the efficiency of germination just by giving them kind of a mock or mimic some sort of winter. I will say that not always is it about cool, moist conditions. It's very species dependent, and some species have more than one stratification need or requirement. Some have a double dormancy where they need both stratification and scarification, or scarification and stratification, or warm stratification first with a cool stratification later. And that's essentially what I'm showing with this peony seed here. So if you had peonies that were species peonies, and most of our peonies are not, they're hybrid, and you wanted to grow them from a seed and germinate them, you would first have to have a warm stratification period so that the embryo could completely develop. The radicle would then emerge just a little bit, and then it would need a cold stratification period because the epicotyle, which is that initial stem, also has another dormancy period. That means it needs to go through winter. And several trees will have that initial germination, or the radicle will emerge in the fall, and then it'll sit there and wait for kind of a cold period as well, or winter, before it sends up a shoot. And that's what's happening here with peony. So warm stratification, then cold stratification to actually get it to germinate. I don't want you to confuse pre-chilling with stratification. This is a great, excellent article that was by Garden Gate magazine a couple of years ago on how to pre-chill different seeds that you purchased or that you harvested off of something else. I think these were delphinium seeds. And they basically put these seeds in ice cubes, froze them for less than a week, so just a couple of days, and then sewed the ice cubes into these different 6-packs. And that pre-chilling method worked really well in helping improve germination for this particular species. This is not a replacement for stratification. So if stratification is needed, that's prior to any other pre-treatments like pre-chilling or imbibing in water for 24 hours before germinating. So certain species have a lot of issues and they need certain requirements. And stratification, if it's required, is usually essential. And nothing else will overcome it except for that 2 months of cool, moist, or 1 month of warm, moist temperatures. This is a great example of what I do when I'm going to stratify seeds. I will collect the acorns or collect the buckeyes. I'll place them in some sort of germination mix or even just peat or something kind of staying moist. If I don't even have that, I'll use some wet paper towels or moist paper towels. I'll put them in a Ziploc bag and I'll put them in my refrigerator for 2 to 3 months. I'll mark on the Ziploc bag when I put them in, and I'll even mark on my calendar about when I need to take them out. This kind of cool, moist condition is ideal. I don't put them in the freezer, I put them in the refrigerator because that 40 degrees is the perfect place for stratification because seed are usually a little bit lower in the soil and may not be experiencing necessarily freezing temperatures at all the time for that entire 2 months. For woody plants, 2 to 3 months is ideal, and if you don't want to research the exact time frame, go for 2 to 3 months. Take out half of them at 2 months and start to germinate them, and if that doesn't work very quickly, you still have the other half, you can try to germinate at 3 months. Scarification is a little bit different. Once again, it's about that hard seed coat. And there's some really fun stories out there about how scarification is essential or important in the germination or breaking dormancy of certain seed. And this tambaloki, I'm not sure if I'm saying that correctly, plant was thought initially to be related to the fact that it was scarified in the gut of the dodo bird. And the fear at the time, this was I think in the late 1800s or early 1900s, that when the dodo bird went extinct, then does that mean that this tree was going to go extinct as well? Because the gut of this particular bird was very rocky, like a rock tumbler in a sense, and really quite intensely needed to break this really hard pit, a lot like a peach pit in a sense. And so it developed into a scarification story of how things will continue as well. So I'm going to ask you a quick question on how you scarify seeds while I plug in my computer, my old computer, which is not liking me at the moment. So if you want to type into the chat how you scarify seeds, what you do to scarify seeds. And please don't put in there that you're going to eat them, because I don't want you to eat them. All right, you're not a dodo bird. Oh, I like it. Nail clippers, that's a great idea. And water, good. Sandpaper. Oh, excellent. So here's what I want to show you on what we do. Ah, I like it. Sand and shake. That's a good idea. So here's how we typically scarify seeds. There are mechanical methods which are a lot like that sandpaper or a file that you're seeing in this picture, or the nail clippers or pruners. Sometimes even larger seed you can put in a rock tumbler that polish rocks. That works pretty well even with rocks as well. Those are great ways to mechanically damage that seed coat, that hard seed coat, so that it'll germinate. Or we can crack it open so that it will germinate. In this image, we have red bud seed that we're just kind of using, running along a file until we see a slight green of the embryo or the cotyledon, and all we need is a hole. And then we can then place this in water to imbibe real quickly, or even just sow directly into germination media. There are other ways as well to scarify seeds. Mother Nature helps out sometimes with a repeated freezing and thawing cycles. Mother Nature also has fire, so there are some pine cones that only open up and the seeds are scarified after they're burnt in a sense. I would tell you that that's probably not a technique I would use, and that's same with an acid soak as well. There are some seed that, as a horticulturist, if I want to germinate them, I might soak them in sulfuric acid. That's generally not something I recommend because it's a little bit tricky and not something most homeowners would do. So stick to the mechanical methods, but know that sometimes there are some super hard seed coats out there that will even survive soaking for 30 minutes in sulfuric acid. Once again. Excellent. So once again, priming seed is not necessarily breaking a hard seed coat. It's just allowing that seed to get ready after it's already has its dormancy requirement met. I will also say not every seed has a dormancy requirement. Generally, temperate things do, but your vegetable seed, an annual seed, may not have a dormancy requirement at all. Sometimes we prime seed by just soaking them overnight in water. That's a form of hydropriming. And then if you really get into this, there are other types of priming that you can do, or that some seed companies will actually do, with nutrient priming or salt, matrix priming, where they actually prime them in some sort of hard surface or soil-like material, and they might even prime them with bacteria or fungi. We see sometimes that type of association with priming of seed for certain legumes, encoding them with certain symbiotic materials that help or improve the germination percentage. Once again, very species dependent, and not something that most of us do, but you should know that you can sometimes purchase primed seed, which will greatly improve its efficiency and germination. It's much faster this way. So if you have a fairly difficult soil, maybe purchasing prime seed means it germinates a little bit faster, so it overcomes that need for ideal environment, so that you can go with something pretty quicker as far as growing up and developing. Prime seed, however, doesn't last as long, so that's another thing to remember. So you need to use it all fairly quickly, because it's not going to be something that stores very well. Finally, we're getting to environmental conditions and how we grow things. This is probably where you know the most, and that what's important in germinating seeds is all about having that correct environment. My hope here is that I'll give you a few hints and tips to make it easier. So first decision you need to make if you're germinating seeds is if you're going to direct sow outdoors or you're going to start indoors. With a lot of vegetable seeds, particularly warm season vegetables, we can start them indoors early so that we kind of reduce that growth and development time so that we harvest a little bit earlier. But cool season crops actually prefer almost always being direct seeded outside. So read the packet. That packet or research the seed will often tell you if it's better direct sown outside or if it should be started 6 weeks earlier. You need to assemble the supplies, and there are some tricks and tips on what to assemble as far as supplies for seed starting. Obviously, we want to start with viable seed. You already know what your viability of this seed should be, and if you need to oversow or not, because you've done that germination test or you've purchased fresh seed, and you know that you should get better than 80% germination. You also need to start with some clean containers, some good germination mix. You might think about a heat mat. I'll show you what that is. And then we'll talk a little bit about the water and humidity. And finally, make sure you do a good job of knowing what you're sowing. So make sure things are labeled. You don't have to use plastic plug trays or 6-packs in germinating seed. You can use biodegradable or compostable containers. I've germinated seeds in old eggshells, and I love germinating them in egg cartons as well. You can also make containers in paper or purchase peat pots as well. Remember, when you're using different containers, however, that it needs to be fairly well drained. They can't stay wet even in biodegradable containers because they don't germinate well. And then even with lemon or eggshells, we clean them out first and make them as clean as possible. I'm going to say it's not as important what kind of container you start off with as long as it's clean. I will say it's important to start with a really good germination mix. And I don't want you to use just a regular potting mix. I want you to purchase a germination mix, especially if you've got smaller seed. This mix generally has a little more perlite in it, so it drains really well, and all the components are usually finer than in a typical potting mix. This is ideal because then the seeds don't filter or get lost in the potting mix. So a germination mix might be a little more expensive, but often is worth it. And purchasing new germination mix every year means you're starting off with something sterile or fairly sterile so that you have less issues maybe with damping off. Soil temperature is also important in this instance as well. And it's not outdoor soil temperature. This is more indoor soil temperature and how that germination mix is maintained. And with this one, a heat mat is really important. So this is another one where it might be an expense, a fairly big expense, because sometimes these heating mats are $100, but it's worth the effort. Because if you see over here on the chart, if you are sowing seed and germinating at a proper soil temperature based on what's needed and can maintain 70 or 77 degrees, you can cut the germination time in half for some species. So, and 70 is usually our target, 70, 72 degrees for most seed. And sometimes having that heat mat is perfect. And these were created to be underneath a seed flat and get wet and not electrify everything, even though they're plugged in. Water and humidity are also equally as important. I always start my seedlings with a humidity chamber over them. So I've got the soil moist, I've got the heat mat underneath, and I've got this kind of humidity dome over this. You can spend money on this, or you can just have even a Ziploc bag that will kind of keep that humidity in. In the greenhouse here at Iowa State, we have a misting system, so that keeps that humidity high. Once again, just keeping it high means you're watering a little bit less, but it's also the ideal conditions for seed to kind of imbibe and start to germinate. Some seed are very specific on their environments that they need to make this happen, like orchid seed. Orchid seed doesn't often contain an endosperm or cotyledons, so they have to have the perfect nutrient, water, humidity situation to actually germinate in the wild. And that's why most people who are germinating are attempting to germinate orchid seed are actually doing it in agar and baby food jars or tissue culture. Once the seeds start to germinate, or if you need to water as the seed are germinating, you want to use something super soft and small and delicate. Because remember, these seedlings are very delicate. So you have a couple of options here. You can purchase something like a fogger, which goes on the, it's a breaker that can go on a water hose and just kind of creates this really dense fog or mist, which is perfect for seedlings. You can use a misting bottle as well, or these kind of hand waterers for seedlings. So you can purchase something if you want, or you can just water from below and allow it an hour or so to soak up. Any of these are ideal. We just got to make sure we keep them consistently moist. We don't leave it in the water saucer of water for more than an hour to soak up. That's staying a little too wet. We want to avoid something called damping off. Damping off is a series of fungal pathogens that once they hit, they kind of attack the seedling at the soil line and it spreads throughout a flat fairly quickly. You can lose everything in 48 hours. So keep away from staying too wet, and then try to stay not too dry, because both somewhere in the middle is where you want to stay. And if you're too wet on your seedlings, damping off frequently happens. Light's not needed for seed germination, believe it or not. Lettuce, maybe some spinach will require light to germinate. Everything else really will germinate without it. Light, however, is essential for plant growth and development, for the seedling growth and development. And if I'm starting seed indoors, I'm starting them under some sort of fluorescent or LED lights. These lights are usually very close to the plant, so I'm going to put them within 6 inches of the top of the seedlings so that I can make it as intense as possible once they've started to germinate, because I want my seedlings to be as compact and as sturdy as possible. I might even put a fan in the area to simulate some wind and force a little bit of movement. I'm not giving them a windswept appearance. I'm just forcing some air over there so that they're moving a little bit so that they develop a sturdier stem. And then once again, damping off is still an issue. So moist, but not wet. Before I take seeds outside, I'm going to acclimate them. And this is also equally as important, and that's just about increasing exposure to sun and wind and cooler temperatures in the spring. This can take a little while, so tell me, how long do you think this will take? Just a few of you quickly in the chat, what do you think? How long should you spend in acclimating? I've got generally a week to 2 weeks. That's ideal. The more you can acclimate them, the better. Often depends on the temperatures outside. You need at least 5 days. You're just slowly exposing them to more light, cooler temperatures. The first couple of days you take them out, you bring them back in at night. Then by the last few days, maybe you're actually taking them out to where you're going to plant them, and leaving them there overnight. Yeah, very important, and usually a great way to reduce any transplant shock. And I've only got a couple of minutes more and only a couple of more slides. So when you're done with all of this, sometimes we want to save seed from things that have done really well. And once again, I'm going to remind you to make sure that it's true to type if you're going to save seed. Hybrid seed, you don't necessarily know what you're going to get from that. Once you harvest the seed, you need to kind of get the seed from the fruit. So sometimes that usually means cleaning up the seed, whether it's dry seed and just kind of breaking it apart so that you actually find the seed itself, or maybe even fermenting the seed in buckets of water. And then the seed that float never are considered viable. We dispose of those because they're often hollow. It's the seeds that stink and they're in all the mucky stuff that you want to kind of get out and dry and try and save. Once you get them to, you have the individual cleaned seed and dry seed, we often store them in a cool, dry, airtight containers in the dark. And this is where you can store them in the freezer if you want, or you can store them in a refrigerator. It's kind of up to you. Read a little something about those seeds and you can figure out how long they will store and what the best storage conditions are. And then finally, there's some really weird seed out there that don't go through the typical processes, that don't go through the formation of the union of the gametes. And that are kind of cool to maybe start off with if you want to do something weird. They're called apomictic or apomixis seed, and citrus is often apomictic. So it produces seed, but it's from the maternal side of the plant. So it's actually a clone of that parent. So you can grow an orange that looks a lot like the orange that it grew on in Florida or California. Kind of weird. So always have fun when growing different seeds or playing around with your plants.